Hello everyone, I'm Janani from Government Science College, Bangalore. I'm doing my Masters in Biotechnology and I'm going to present to you about homology modeling of neuromedin type 2 receptor using the software modeler. Neuromedin type 2 receptor, which is also called NMUR2, is located in the hypothalamus region of the brain, and the main function of this receptor is to bind to neuromedin U and regulate food intake and body weight in animals and humans. There are three important steps in homology modeling. The first step is the identification of a suitable template. Next comes the alignment of the template sequence and the target sequence, and then based on this alignment we try to predict the structure of the protein. In this case it is NMUR2. This is the homology search result performed by modeler software. These are the top 5 hits that are closely related to our target sequence. Out of these we chose 4BUOA chain of neurotensin type 1 receptor of rat as our template because it has a better resolution of 2.8 angstrom compared to others. And next step was to align the template sequence and the target sequence. Through this we commanded the modeler to perform a pairwise alignment based on the defined scores and cap penalties. This is the result of the alignment, and we can see that there is asterisks indicated below some of the columns. These asterisks show that these residues are conserved over the two sequences. Now, based on this alignment, we have to predict the structure of the protein NMUR2. To do this, we commanded the modeler software to construct 3 models based on the alignment, and these are the 3 models: model 1, model 2, and model 3. And this is a log file containing the summary of all 3 models with dope score and GA341 score. Ideally, the model which has the least dope score is considered to be as the best model. Here, in this case, we can see that the first model has the least dope score of minus 46,000 to 76.33, and so. Therefore, we declared that model 1 is the best model for NMUR2. On structural analysis of this model, we see that it is a typical GPCR with 7 transmembrane alpha helices connected by loops. And we can also see that there is a small stretch of anti-parallel beta sheet in between 4th and 5th helix. This model was validated by plotting Ramachandran plot. Ramachandran plot for this protein showed that 92.7% of the residues lied in the most favored region, 5.6% lied in the allowed region, and only 7 residues lied in the outlier region, which indicates that the overall quality of the protein is good. Now, the importance of modeling this NMUR2 receptor is that it is claimed as a potential drug target against obesity based on the experiment conducted by several scientists. In these experiments, neuromedin U was injected to the cerebrospinal fluid of obese mice. The neuromedin U bound to the NMUR2 receptor and triggered signaling responses, which ultimately resulted in reduced food intake, reduced body weight, increased locomotor activity, and increased body temperature in these obese mice. This overall results indicate that neuromedin U is a potential appetite suppressor when combined with NMUR2. So therefore, we conclude by saying that NMUR2 receptor is a potential drug target against obesity, and we also would like to add that agonists that are specific to neuromedin U2 receptor could act as a potential anti-obesity drugs. Thank you for the opportunity.